Good morning, everyone. We'll be talking about surface plasmon resonance based drug discovery. And these are the areas we'll be covering in our presentation. So, the inspiration behind this topic is a drug discovery company named as Beatica. It is located in Uppsala and it uses SPR biosensors to discover the new small therapeutic agents. It uses the SPR data to study the interaction between molecules. So I'll be talking about the principle and instrumentation part of the SPR. So its main principle is total internal reflection. So when an incident light, it propagates through a medium of denser reflective index to a medium of lower refractive index, it shows reflection rather than refraction. So next is, here we have a very simple presentation of its instrumentation and working. So first, I'll start with the instrumentation part. Instrumentation part, it includes a light source, and this light source can be a light-emitting diode. And in this example, for light-emitting diode, we generally use the wavelength of infrared region. Next, we have a prism. And next component is the biosensor, and this is the most important part of SPR. This biosensor is a glass slide, and it is very finely coated with gold metal. And on one side of the gold, we have immobilized ligand attached to it. And for this example, we have shown antigen and antibody interaction. Next, we have sample chamber. In this sample chamber, we inject a sample containing analytes, and then the detector. So what exactly happens is, when an incident light from the source, it enters into the prism of denser medium, of denser refractive index, and it is focused on the lighter, denser refractive index of the biosensor. It shows total internal reflection, as shown, like you can see the reflected light ray. So what happens exactly in biosensor chip? Due to the absorption of photon, the electrons get excited, and the oscillation produces surface plasmon. And this surface plasmon is basically sensitive to the refractive index, change in the refractive index near the biosensor. So this change in the refractive index is observed when we inject a sample containing analyte. So this sample, when it interacts with the ligand, so the binding occurs between the antigen and antibody, and because of this binding, we observe change in the refractive index. And this change is further detected by the detector. So the data from the detector, it looks like this. This is called as sensogram. So in the y-axis, we have binding response, which is plotted against time. So the baseline represents the stable form, that is, all the ligands are free, and it is the stable form. And next is the when we inject sample, and there's interaction between the antigen and antibody, or say the ligand or analyte, and this represents a shoot in the graph. Then we have equilibrium. Equilibrium is basically when the binding of antigen and antibody is like the association and dissociation is equal. Next, we have dissociation of antigen and antibody. So the most important thing about SPR is it's a label-free technique. So there are no washing steps involved in SPR. So basically, dissociation is done by changing the pH of buffers, pH of the sample. And then we have the baseline, and then the next baseline represents the regeneration of the next cycle. And beyond, we'll be talking about the application part.
So now that we know what SPR is and how it works, we can talk about how to use it. As you see on the graph here, we can study the binding kinetics over time. So that's a great way to study ligand binding. you can use it to screen for drug leads when studying different proteins, as we shall see. A good example of this is to use receptors, because they are already ligand activated, so there's a binding pocket that you can use. And if you can get a good drug that is competitive with the native ligand, you can get competitive binding, and if the affinity is higher, you have a really good inhibitor. You can also develop cancer drugs. As you probably know, cancer is caused by the faulty regulation of the cell cycle, leading to uncontrolled growth of the cells. So if you can interrupt the signaling pathway in the cell that causes cell division, then you can stop it and hopefully prevent cancer from growing further. Ideally, you would have an inhibitor that binds covalently to the protein, killing it off. So if the inhibitor disassociates, it returns to the pool of protein and can continue to work. But if it's irreversible binding, it's just gone. An example of this approach used is with LSD1, which we'll talk about in detail in a moment. There are several advantages of using SPR. For example, you can see the molecule concentration, you can track the binding kinetics, and you can see the specificity of the different ligands. You can see the relative binding pattern, or cooperativity, meaning how the ligand affects the protein, if there's a conformational change or such. And as Vaishali mentioned, it's a label-free method. You don't need to add a GFP to the protein. You don't need anything that glows in the reaction, since it measures the interaction in a different way. So there's no steric hindrance. It's just the native protein and the ligand, which is great because it mimics the physiological environment. There have been some advancements in the technology since it was invented, mainly pertaining to the amplification of the signal. For example, if you have a very low concentration of the protein, then you can get a higher response still. How you do this is basically you have nanoparticles, typically of gold, that's coated with the ligand. And when it binds, because there's a gold particle there, there's a much bigger mass, which gives you a higher read than normal, amplifying the signal.
Okay, I will be just a second. Not really handy. Okay, so I will tell you about the case studies that we have worked on. So our inspiration was Beatica's work and how they used SPR, and they have published many articles, and we focused on the two topics, and one of them is LSD1 inhibitors. And this molecule is a part of Beatica's oncology pipeline. And another one is NNRTIs, which stand for non-nucleoside reverse transcriptase inhibitors. So what is LSD1? It's not a drug to start for. It's a histone demethylase, and it's involved in histone 3 protein and lysine demethylation, specifically lysine on position 4 and position 9. It can act as both co-activator and co-repressor, which is different in comparison, for example, to acetylation, which is usually linked only to gene activation. And the catalytic reaction occurs in the areal domain, which is pointed here in red.
So why is LSD1 important? LSD1 has found to be overexpressed in many human cancers, as lung or blood cancers, pancreatic cancers as well. So this is the list of currently developing LSD1 inhibitors. They all target different kinds of cell cycle, and some of them are already in phase 2 clinical trial. And the ones that you see in red, they have all been developed from trianylcypramine, which is also the first inhibitor that was developed for LSD1. Also, as you can see, like out of 4 of these, 3 are targeting leukemia, and EML stands for acute myeloid leukemia. And also Alzheimer's disease is targeted with LSD1 because LSD1 is also involved in some kind of transmission of neuron signals inside of the cells. And those are also all irreversible inhibitors. Reversible inhibitors have been developed from different kinds of molecules and chemicals, and they also target different kinds of cancers. So Beatica's goal is to develop a new inhibitor, and they have been focused on developing a reversible inhibitor that will target differentiation of leukemia stem cells in AML. So this is an example of one of their works. They have used SPR to screen their library, their fragment library, and they have been decreasing the number of hits and increasing the properties of the chemical. And they have ended up with 4-sanol-phenylglycinamide, which has IC50 of 0.083 micromolars and KD of 0.032 micromolars. And another study that we have focused on is HIV-1-RT. So this is an enzyme that is involved in the main part of HIV in being inserted into humans. So it has 2 main functions. One is RNA-dependent DNA polymerase, where this part of enzyme synthesizes strand that is going to be the complementary to the virus. So that's a minus strand. And then DNA-dependent DNA polymerase is going to synthesize complementary strand, and this DNA molecule is later on going to be inserted into the host. 2 types of inhibitors have been developed for this enzyme, and those are the nucleoside reverse transcriptase inhibitors and the ones I have already mentioned, non-nucleoside reverse transcriptase inhibitors. So these chemicals here that you see are the 3 currently approved FDA NRTIs that's cures for HIV. So usually they have the resistance develops really quickly, and they have to be used in a combination for treatment, and some of the side effects can be really severe. So the process of finding the cure for AIDS is still developing, and what Beatica did is they have been focusing on both finding a new inhibitor and also finding a way to see how to maximize the potency of the inhibitor. So they have proved the importance of the slow dissociation and the relaxation state of antiviral efficacy. Also, they have found a new promising inhibitor, which is bromindanone. So that would be all from us. So as a summary, we have gone through SPR, and SPR provides the high specificity, label-free, and real-time detection. It can be used for many things, and one important thing is the drug screening to target small molecules. That's how they ended up developing LSD-1 as a potential drug target and developing novel HIV-1 RT inhibitors.